Good morning, all of you.
Good morning, sir.
Good morning. Okay, we are discussing about the plasmolysis. Okay, to understand the plasmolysis, first of all, we have to understand some concepts like what are the different types of solutions. Okay, that is isotonic solution, hypotonic solution, and a hypertonic solution. Okay, that is if the concentration of the cytoplasm of the cell is equal to the concentration of the external solution or external environment, then we say that the cell is in isotonic with the external environment. And next, if the concentration of the external solution is low when compared to the cytoplasm of the cell, then we say that the cell is hypotonic to the the cell is hypotonic to the external solution. Okay, and next, if the external solution is having a higher concentration, okay, if the external solution is having higher concentration than the cytoplasm of the cell, then we say that the external solution is hypertonic to the cell. Okay, that is, and what happens when these conditions will occur? Okay, in isotonic solution, the net movement of water molecules is 0. So we observe there is no change in the cell. Okay, size or concentration of the cell. But when when you observe, when you place the cell in a hypotonic solution, there is the solution, the external solution, which is having lower concentration than the cytoplasm of the cell. Then what happens? Okay, because of the difference in the potential, okay, the water continuously enters into the cell. As a result, the cell will swell and finally lead to the bursting of the cell and finally leads to the death of the cell when the cell is placed in a hypotonic solution. At the same time, okay, when you place the cell in a hypertonic solution, then because of the difference in the water, okay, water potential, the water comes out of the cell and enters into the external environment or external solution. As a result, the cell will shrink. Okay, the cell, okay, that is the cytoplasm of the cell will loses and the cell will develop certain wrinkles, which we call it as the shell is shrinking. And this phenomena we are calling it as exosmosis, the movement of water out of the cell into the external environment. We are calling it as exosmosis. It occurs when the cell is placed in hypertonic solution. At the same time, when you place the cell in a hypotonic solution, the water will enter into the cell, that is called endosmosis. So in isotonic, we don't observe any resultant change, but in a hypotonic solution, the endosmosis will occur. In a hypertonic solution, the exosmosis will occur. And next, the plasmolysis. Okay, what is mean by plasmolysis? Okay, when you observe the plant cells, okay, when you observe the plant cells, okay, the plant cells along with the plasma membrane, surrounding the plasma membrane, will find another outer covering, which we call it as a cell wall, which is a tough, rigid, and do not allow the plasma membrane to extend beyond its limit. Okay. So what? Okay, as it is rigid and tough. Okay. Then what happens whenever you place a animal cell in a hypotonic solution? The cell will absorb water, or the cell will intake water by endosmosis, and up to the flexibility of the plasma membrane, it can withstand. If the water enters beyond its flexibility of the plasma membrane bursts, leading to the death of the cell. But that is not happens with the plant cell. Okay, whenever the plant cell is placed in hypotonic solution, okay, the water will into the cell by endosmosis. Slowly, the plasma membrane will increase and get pressed with the cell wall. That means it get attached with the cell wall, and up to the extent of the cell wall only the plasma membrane will increase. Beyond that, the plasma membrane will not extend. Okay, that is and so there is no bursting of the cell in case of plant cells when you place a plant cell in hypotonic solution. But when a plant cell is placed in a hypertonic solution, when a when a plant cell is placed in a hyper solution, what happens? Slowly the water will move out of the cell by exosmosis. Okay, slowly the cell loses water by exosmosis. Okay, as a result, what happens? The pressure, okay, the osmotic pressure will be gradually decreased. Okay, as a result, what happens? Slowly the plasma membrane initially the plasma membrane get separated from the cell wall. Okay, if the exosmosis is still continued, okay, the plasma membrane will develop certain wrinkles and the plasma membrane of the cell will shrink, okay, as it moves away from the cell wall. Okay, there is a plasma. Okay, this phenomenon of development of wrinkles are okay, the plasma membrane, the shrinking of plasma membrane. When you place a plant cell in a hypertonic solution, is nothing but called as the plasmolysis. Okay, this phenomenon of development of wrinkles or shrinking of a plasma membrane after separating from the cell wall, when the plant cell is placed in a hypertonic solution, and this phenomenon we are calling it as a plasmolysis. Okay, that is what we can see here. Plasmolysis occurs when water moves out of the cell and the cell membrane of the plant cell shrinks away from its cell wall. That means initially the plasma membrane separates before it is very tightly pressed towards the cell wall, but once is losing water from the cytoplasm, gradually the osmotic pressure will be decreased. As a result, the plasma membrane get separated from the cell wall. Okay, get separated from the cell wall, and next develops wrinkles or it shrinks. Okay, that is what we see here. Okay, when this will occur means this occurs when the cell is placed in a solution that is hypertonic to the protoplasm. Okay, when a plant cell is placed in a hypertonic solution, okay, because of the difference in the water potential, the water will move out of the cell. As a result, the cell membrane gets separated from the cell wall and finally develops string, okay, and develops wrinkles and strings. This phenomenon we are calling protoplasm. This occurs because of exosmosis in the plant cells. Okay, water moves out. It is first lost from the cytoplasm and then from the vacuole. Okay, we all know very well. Okay, the osmotic pressure of the plant cell is mainly depending upon the vacuole. okay, depending upon the vacuole, okay. And so when you observe here, so here, first when a plant cell is placed in a hypertonic solution, first the water is lost from the cytoplasm, and later the cytoplasm takes up water to maintain the normal condition. The cytoplasm will take water from the vacuole, and that one also will be gradually undergoes exosmosis. So first the cytoplasm loses water, and then it will loses water from the vacuole. So the water then withdrawn out of the cell through diffusion into the extracellular fluid causes the protoplasm to shrink away from the walls. Okay, due to the movement of the water out of the cell by exosmosis. As a result, what happens? It causes the plasma membrane to get separated from the cell walls, and it develops certain okay, and the plasma membrane develops certain wrinkles, and okay, and forms as a shrink. And this is okay, and now the cell is said to be plasmolized. The cell is said to be plasmolized, and this phenomena we are calling it as a plasmolysis. The movement of water occurred across the membrane, moving from an area of high water potential to an area of lower water potential outside the cell. Okay, here, okay, all this phenomena is based upon the osmosis only, because of the difference in the concentration of the cytoplasm and the external environment. As the external environment is having higher concentration than the cytoplasm or protoplasm of the cell, what happens? The water will move out of the cell, okay, from higher water potential to the low, okay, region of external solution where water potential is low. So it is following the phenomenon of osmosis. Okay, there is okay. What occupies the space between the cell wall and the shrunken protoplasm in the plasmolyzed cell? It is not having any material. It will be material, or it is filled with water, because the cell wall is permeable to all the substances. Here it is not involved in osmosis. So the external solution, the cell wall, okay, that is the space between cell wall and the plasma membrane is filled with the external solution only. Whatever the solution in which the cell is present, okay, that solution only present between the space between the cell wall and the plasma membrane, because the cell wall is permeable to all the substances. It is not a selectively permeable membrane. So as it allows all substances, okay, so the cell wall, okay, the space between the cell wall and the okay, cell, okay, and cell wall and plasma membrane is filled with external solution. Okay, that is what occupies the space between the cell wall and the shrunken protoplast in the plasmolyzed cell. Okay, it is nothing but the external solution. That is a hypertonic solution that is present in the external solution. That one only fills the gap between cell wall and the plasma membrane, because the cell wall is permeable to all the substances. It is not a selectively permeable membrane. And next, okay, what happens when the cell is placed, when a plasmolyzed cell is placed in isotonic solution? So when the cell is placed in an isotonic solution, there is no net flow of water towards the inside or outside. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. Okay. That means if the net movement of the water molecules is 0, or when the cell is in equilibrium, okay, when it will attain means when a cell is placed in isotonic solution, the cytoplasm of the cell and the external solution are in equilibrium. So the net movement of the water molecules is 0. At this point, the cell is said to be flaccid. That means when the cell is when a cell develops flaccid nature means when a plant cell is placed in isotonic solution. Okay, at this point, the cell will be in equilibrium condition. The cell will be in equilibrium condition. Okay, as a result, the net movement of the water molecules is 0. That means how many number of water molecules are entering into the cell? The same number of water molecules will move out of the cell at this equilibrium point. At this point, the cell is said to be flaccid. And next, the process of plasmolysis is usually reversible. Okay, after, okay, when you, okay, that means the plasmolyzed plant cell can be reversed. That means it we may, okay, that means we can change the plasmolyzed cell, plant cell into normal cell. How means, how it is achieved means, okay, when the cells are placed in a hypotonic solution, that is from the region of higher water potential or okay, in the region of higher water potential or dilute solution as compared to the cytoplasm, water diffuses into the cell causing the cytoplasm to build up a pressure against the wall. This is called a turgor pressure. So when you observe here, okay, when a plasmolyzed cell is placed in a hypotonic solution, that is the solution which is having a lower concentration than the cytoplasm of the cell. Now the water will enter into the cell through plasma membrane. Through plasma membrane. Okay, and gradually the wrinkles are removed, and slowly the plasma membrane distends and finally get pressed or get attached with the cell wall. But here we are not placed the cell in isotonic solution. We have placed in a hypotonic solution. So that means we have placed in a solution which is having low concentration than the cytoplasm of the cell. So continuously the water will try to enter. But because of the continuous water is entered into the cell, what happens? That okay, the some pressure will be applied on the plasma membrane by the water. Okay, and whatever the pressure that is exerted by the water on the plasma membrane, the same pressure will be applied on the cell wall. In return, as the cell wall is rigid and tough, okay, it will resist to the pressure exerted by the plasma membrane. In return, it will exert the same pressure in the opposite direction. Whatever the pressure that is exerted by the water on the plasma membrane, the same pressure will be exerted by the cell wall on the plasma membrane. And this pressure, we are calling it as a turgor pressure. Okay, that is so that is what he is saying here. Okay, what is meant by turgor pressure means the pressure that is exerted by the cytoplasm. Okay, cytoplasm or plasma membrane on the cell wall. Okay, that pressure we are calling it as the turgor pressure. Okay, that is okay, this turgor pressure, nothing but the pressure that is exerted by the plasma membrane on the cell wall, and in return the cell wall on the plasma membrane, because both are same but in opposite directions. Because as the cell wall is rigid and okay, and tough. So that's what we can see here. So, okay, the process of plasmolysis is usually reversible. That means we can change a plasmolysis to a normal cell. How means? By placing the cell in a hypotonic solution. Okay, as a result, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall. Because if you place isotonic solution, there is no much pressure. But as we are placing in a hypotonic solution, so the the cytoplasm will exert some pressure, or the plasma membrane will exert some pressure on the wall or cell wall. This pressure we are calling it as a turgor pressure. The pressure exerted by the protoplast, here the protoplast means that is cell membrane, due to the entry of water against the rigid walls is called pressure potential. Okay, whatever the pressure that the plasma membrane that is exerting on the cell wall, that pressure we are calling it as a pressure potential. Because of the rigidity of the cell wall, the cell does not rupture. And this turgor pressure is ultimately responsible for enlargement and extension growth of the cells. That means in plant cells, okay, this pressure, okay, that is the pressure exerted by the cytoplasm on the cell walls, is mainly responsible for the growth of the plant cells, enlargement and growth of the plant cells. Okay, that is so what would be the pressure potential of a flaccid cell? Okay, in a flaccid cell, no pressure potential, no turgor pressure, because the cell, okay, the cytoplasm of the cell and the external solution are in same concentration. So we don't find any development of turgor pressure in a flaccid cell. When you place a cell in a hypotonic solution, then only entry of water into the cell is observed, and that water will exert some pressure, or the cytoplasm of the cell will exert some pressure on the cell wall, and that is called as the turgor pressure. So the flaccid cells which are placed in hypotonic solution, which are placed in isotonic solution, do not exert any turgor pressure or do not develop pressure potential. Okay, which organisms other than plants possess cell wall? Can you name any any organisms which possess cell wall other than plants?
Sir, Munira, bacteria.
Okay, bacteria, and next fungi. Okay, bacteria and
Protista,
fungi.
sir.
Okay, the fungi also having the cell wall now made up of chitin.
Yes, sir.
Okay,
Protista also have, sir.
Asma. Okay,
Protista, sir.
unicellular plants. Okay, that is photosynthetic protist. You say. Okay, photosynthetic protists are having the cell wall. Okay, photosynthetic protist, and next bacteria, and next fungi are having the cell wall. In those organisms also we'll find, okay, okay, someone is saying mollusk. Okay, mollusk is an animal, man. Okay, it is having shell, not cell. Okay, cell wall. The total individual cell is not covered. The total body of the organism is enclosed by a hard calcareous shell. Individual cell is not covered by the cell wall. Okay, na? So molluscans do not have cell wall. They have got a shell. Okay, that is about this plasmolysis. Understood, all of you? Okay, all of you understood what is a plasmolysis cell, what is a flaccid cell, when the turgor pressure is developed. Okay, what is meant by pressure potential? Okay, which is responsible? Okay, this pressure potential only is driving the water to move upwards. This pressure potential only helping the cell to grow. Okay, which helps in the enlargement of the cell. Okay, all these are because of the pressure potential. That is the pressure that is exerted by the plasma membrane on the cell wall due to the continuous entry of water into the cytoplasm of the cell. Okay. Okay, that is. And next, the syllabus for the Monday's test. Already I informed you. It is the unit 3 complete. Cell, the structural and functional unit of life. And next, biomolecules. And next, the third one, that is cell cycle and cell division. All the three together will have the test on a Monday. Anybody is having any doubt regarding this?
No, sir.
Okay, now. Okay, unit 3 complete. Already I shared the materials regarding this. You can practice those questions. If you have any doubt, you can send the message to my WhatsApp number. Okay.